Hello, my name is Sветlana Sokolova. I am presenting my research on the COMT rs4680 gene in addicts of synthetic cathinones and cannabinoids. This work was carried out at the Institute of Cytology and Genetics, Siberian branch of the Russian Academy of Sciences. The relevance of this study is due to the growing use of synthetic drugs. According to many studies, the use of cannabinoids and cathinones causes more serious negative effects. The scientific novelty of the work lies in the fact that for the first time, an analysis of the rs4680 polymorphism of the COMT gene was carried out in people dependent on synthetic drugs. The aim of the work is to study the COMT gene and selected polymorphism in synthetic drug addicts and healthy individuals. Objectives of the work: conduct a comprehensive bioinformatic analysis, conduct a comparative analysis between addicts of synthetic drugs and healthy people, between individual groups of addicts, and conduct mathematical processing and analyze the obtained data. Why did I choose this gene and polymorphism for my research? Catechol or methyltransferase is a gene encoding an enzyme responsible for the metabolism of catecholamines. Changes in its function can affect the levels of these neurotransmitters in the brain. There are two isoforms of the COMT gene, soluble and membrane-bound. MB-COMT is the membrane-bound form of COMT, which is associated with the membrane and active in the brain. The MB-COMT gene was used because it better reflects the impact of rs4680. The rs4680 polymorphism is a singular nucleotide polymorphism affecting the cognitive function and emotional state of a person. In homozygous carriers of the AA variant, the rate of a dopamine catabolism is 3 to 4 times lower than in carriers of the G variant. The above data were obtained through bioinformatic analysis of the rs4680 polymorphism. The POLYPFAN2 analysis classified the mutation as benign with minimal impact, while SIFT confirmed that the function is preserved. Additionally, 3D modeling with PyMol demonstrated differences in the protein structure for wild-type and mutant forms, highlighting the influence of the substitution on the configuration of the active center. Methods used: bioinformatic tools and databases, polymerase chain reaction, restriction enzyme digestion, electrophoresis, and statistical methods. Study populations. The polymorphic DNA loci of 165 people addicted to synthetic drugs and 150 healthy people were studied. All subjects were male, living in the West Siberian region. The average age was 25 years. During the study, I performed genotype isolation in a total of 315 individuals using the polymerase chain reaction. Subsequently, restriction analysis was performed, which included restriction enzyme digestion and electrophoresis. Next, the obtained data were analyzed using statistical methods, including the Hardy-Weinberg law, law of population equilibrium, the chi-square test, a criterion for testing hypothesis about the distribution law, and other methods are presented on the slide. Moving on the analysis of the obtained results, genotypes and alleles were calculated in percentage and quantitative terms. Based on the data from Table 1, the frequency of the rare AA genotype was slightly higher in the addicted group compared to the healthy group. However, the frequency of the rare A allele was nearly identical between the addicted and healthy groups. Upon comparison, the following chi-square value and p-value were obtained, indicating that the addicted and healthy groups do not significantly differ. The Hardy-Weinberg equilibrium was then tested for each study group to verify the validity of the observed differences between the groups. Based on the data in Chart 1, no statistically significant differences were observed. This data previously interpreted in the preceding figure along with the chi-square and p-values also support this conclusion. Therefore, the Hardy-Weinberg equilibrium is maintained. To further confirm this statement, a visualization was generated using Python and Matplotlib. Then, the group dependent on synthetic drugs was divided into three subgroups: patients who used only cannabinoids (SC), who used synthetic cathinones (SP), and patients who used both synthetic cathinones and cannabinoids together, mixed group. Based on the data in Table 3, the highest frequency of the rare AA genotype among patients was found in the mixed group. The highest frequency of the rare A allele was observed in the group dependent on synthetic cathinones. The conducted analysis of the frequency of psychosis in drug addicts revealed 26 people and their genotypes. It is evident that the SP group has a higher number of individuals with mental disorders. This supports the notion that synthetic cathinones, due to their more harmful chemical and fully synthetic composition, differ from cannabinoids. Additionally, it can be concluded that 21 individuals with one or two A alleles in their genotype, accounting for 80% of all dependent individuals with identified mental disorders, provide evidence to confirm that the A allele influences predisposition to psychosis. Moving on to the conclusions that you can see on the slide. Thus, we have identified for the first time frequently occurring haplotypes that can be considered potential regulatory markers in the development of dependency on synthetic psychoactive substances. Future bioinformatics studies may employ machine learning to predict dependency risk based on COMT genotypes and haplotypes or conduct genome-wide association studies to identify additional genetic markers. This slide presents a list of sources I used in conducting my research. That's all. Thank you for your attention.